Okay, class, so we're on part two now, and mainly in part two, we're going to cover the bioenergetics of it. And so to start off in its sort of most sort of global look at how the whole uh, ATP synthesis works is this thing called chemiosmotic coupling, where basically high energy electrons get transferred down an electron transport chain, and eventually, in the case with mitochondria, will end up on oxygen. And in the process, what that does is it pumps protons into the intermitochondrial space. And then you have your ATP synthetase, and the protons then can flow down through the ATP synthetase, and that actually converts ADP into ATP with some inorganic phosphate. So this electrochemical proton gradient is how we basically synthesize ATP. And we're going to look at how this all functions in pretty great detail. But in its most simple form, if you can basically take sunlight or foodstuffs, and you can get high energy electrons out of that. And then using those high energy electrons, you can establish a transmembrane electrochemical proton gradient, and that electrochemical proton gradient can drive ATP synthesis, or as we saw before with the membrane potential, it helps with membrane transport, and also it can sort of cause bacterial flagella to rotate. Now, we already covered glycolysis, and that's basically the conversion of sugars to pyruvate. Here's sort of the schematic. We did it sort of early on. And once again, this is not a course in biochemistry and or a course in metabolism, so I don't necessarily need you guys to memorize this, but I do need you to sort of appreciate that one molecule of glucose with 6 carbons is converted into 2 molecules of pyruvate with 3 carbons. And in this whole process, 2 molecules of ATP get consumed, and that produces 4 molecules of ATP and 2 molecules of reduced NADH. Okay, and this is where some of the energy in glycolysis comes from. We've also covered this in quick review, oxidation reduction. This is something that I consider really important. I really want you guys to know and appreciate this. So basically the reduction is the take-up of electrons, and oxidation is sort of the breaking of bonds, okay? And so actually oxidation is the loss of electrons and reduction is the addition. So wherever the electrons are going, protons generally follow, so it's oftentimes easy to tell what's reduced and what's oxidized, right? So this molecule is reduced because it's got extra protons, which means it has extra electrons, and this is oxidized because it has a double bond there, right? So if you sort of remove the electrons from here, they're going to be shared in this double bond. Likewise, in this double bond here, if you reduce it, the protons are going to follow. Okay, and so this is basically how you get NADP in its sort of oxidized form and then in its reduced form. Okay, and here it is sort of a little bit closer. If you look at it, so you have the oxidized form over here, and then in the reduced form, you'll have electrons with a proton attached to it. Okay. So ultimately, NAD plus becomes NADH, and you have 2 electrons and 1 proton in that. We've also looked at the citric acid cycle, where you get acetyl coenzyme A with 2 carbons. It gets fed in and gets added to oxaloacetate to become citrate, and then you go through this whole process, and in it you're producing basically a bunch of reduced NADHs, FADH2 reduced, and GTP. So there's three equivalents of NADH, reduced NADH, one GTP, and one FAD. Okay, and ultimately one molecule of, and this releases actually CO2, 2 molecules of CO2. Okay, if you want to just follow the carbons around. And so ultimately, when you have glycolysis in the citric acid cycle, you take glucose and your sugars and your food sources. They can produce a whole bunch of other metabolites like amino acids and cholesterol and fatty acids and lipids and some other sort of amino sugars and for glycoproteins, as well as free nucleotides, and then heme and chlorophyll, as we'll see, and glutamate and other amino acids are there. Okay, but ultimately, every turn of this citric acid cycle here produces 3 molecules of NADH. Okay, that then ultimately goes on to be used for oxidative phosphorylation. And here's the way it works. You can take your sugars or your fats and your carbohydrates, put it through the citric acid cycle, and that's going to produce NADH with high energy electrons. The high energy electrons are then transferred into the electron transport chain, which basically can then pump protons across the inner mitochondrial membrane to the and pumps the protons to the inner membrane space. And as the electrons flow down, they eventually end up on oxygen, and that is converted to water. And in the citric acid cycle, you're producing CO2. Okay, so basically, when we're using food, we take up oxygen and release CO2 and water. Okay, chloroplasts, it's just the opposite, where you take in some sunlight and then that steals electrons off of water, turns them into high energy electrons, and then they're basically producing a proton gradient. And as the electrons become lower energy, they hit photosystem 1, and eventually those electrons get put on NADPH. Okay, and NADPH and then CO2 are used for carbon fixation, which then can produce sugar molecules, right? So you can sort of think if you had an apple or something, you just sort of are eating the carbohydrates that were made by the plant, okay? And so these are very sort of intimately linked, right? We use plants for food energy and exhale CO2, and then the plants use CO2 and sunlight and release oxygen. Okay, you guys have all had this before, I'm sure, at some point in time in this simplified form, so you pretty much know how it goes. But here's the oxidation of NADH to NAD. Okay, so you have your reduced NADH, and then the electrons are donated, and you basically leave with a hydride ion first and then protons, okay? And these 2 electrons go into the electron transport chain in the membrane. And then you basically get an unstable intermediate and some bond rearrangements, and then you have NAD+, okay? And it's the cofactors in the electron transport chain that all become reduced, and these electrons then can travel through the electron transport chain, which we'll cover in pretty extensive detail in the next coming slides. But here it is, sort of as a global overview. Here's your mitochondria. Basically, it's going to take up oxygen, and you have pyruvate and fatty acids coming in that'll eventually get converted to acetyl coenzyme A, and then through the citric acid cycle, you're producing high energy reduced NADH, and then the electrons are transferred to the NADH dehydrogenase complex, and then off to the cytochrome BC1 complex, and then finally cytochrome the cytochrome oxidase complex, and eventually on to oxygen and producing water. And as the electrons are traveling through the electron transport chain, it's pumping protons into the intermitochondrial space here, okay? And then with this, with all these protons, they flow through the ATP synthase, and in the process what they end up doing is taking ADP and converting it to ATP, and then the ATP is then sent out into the cell, as is the CO2, and ADP is taken up by the mitochondria. Okay, so we'll look at how some of the transport here occurs with these molecules, as well as the electron transport chain, as well as the ATP synthetase. But really sort of have a good global overview of this, how this whole process works, right? And you guys, I'm sure, have had this before in one of your other classes. But ultimately, if you just sort of break it down into its smallest components, you have reduced NADH, plus a half oxygen plus a proton, and then you have ADP plus inorganic phosphate, and then you get oxidative phosphorylation, where you get the sort of oxidized NAD and water, and then that helps to, and that whole process then leads to the conversion of ADP and PI to ATP. So I always give this slide, and it's really disappointing when I'm not here with you guys, because I ask you to questions about the Hindenburg. So the Hindenburg was a Zeppelin made by Nazi Germany in the 1930s, and it was sort of this really famous dirigible, so a blimp. And back in the 1930s, the Germans thought it was a good idea to fill it up with hydrogen, okay? And so this was really kind of a cool aircraft, because it could sort of travel around the world, actually. But it came, and it was sort of dirigible to sort of show off Hitler's might. And he actually flew it at one point in time, not him himself, but the Germans basically came and to New York City, and they needed to dock it, and so they docked it in New Jersey. And at some point in time, when they were docking it in New Jersey, it exploded, okay, because it was basically filled with hydrogen. I want you guys to all get on YouTube and actually look up the Hindenburg explosion, because it was a pretty interesting thing to watch, because it just basically exploded and caught on fire. And one of the interesting things that happened after the Hindenburg exploded, with all this hydrogen and the oxygen in the atmosphere, it got converted to water. And so some of the people that were underneath it, if you watch the video, you can see them running away from the exploding thing. It actually sort of rained on them. But it was kind of interesting. There's some conspiracy theories out there that the U.S., who didn't like the Nazi Germany, basically sabotaged it and blew it up, but I don't know if any of that's true or not. So, but it's interesting to go look at. So definitely go check out the Hindenburg video on YouTube. Okay, so basically when you reduce, get the reduction of oxygen to H2O, here's what happens. You have your protons and your high energy electrons, and they sort of are harnessing the energy, and eventually the protons will end up on, as the oxygen is getting reduced, the protons will go on, and you basically get oxidation of water here, okay? So, or oxidation of, to water. And so the electrochemical proton gradient that exists, that's used to drive ATP synthesis, comes from two areas. There's the proton motor force due to the membrane potential, right? This is the same membrane potential that's driving the signal sequence into the TIM complex. And then you also have a proton gradient, Okay, so it's much more acidic in the inner membrane space than it is in the matrix. And so the natural gradient is for the protons to flow downstream. So between these two things, they're actually driving ATP synthesis. And they're transported by the electron transport chain, right? The electrons that go through the electron transport chain are also pumping protons into the intermitochondrial space here. Okay, there's NAD dehydrogenase, and then the cytochrome BC1 complex, which we'll look at in a second, and then cytochrome oxidase, and eventually the electrons will end up on oxygen, and protons are taken up and created to water. And then all of these protons that are going through convert ADP plus inorganic phosphate into ATP using the ATP synthase. Here's a great schematic of the ATP synthase. It's a F1 ATPase, and you have this little rotor here that actually spins, and the protons from the intermitochondrial membrane, or the intermembrane space here, actually flow through, and that causes the rotator to rotate, and then this produces ADP and inorganic phosphate and converts it into ATP. And here's just the ribbon diagram of this F1 ATPase domain down here. And if you want, I have written down here, it's functions like a biochemical turbine. So if you guys have ever sort of looked at a turbine and it's spinning, you can sort of think of it like that. So transport of molecules uses sort of transport very similar to what we talked about before when we were talking about transporters and ion transport. And in certain instances, you can use the pH gradient to drive transport, like in the case with pyruvate. So this would be a sort of a symporter where you're using the protons to drive the import of pyruvate. Also a symporter to drive in inorganic phosphate. And then you have a voltage-gated ATP-driven exchange here where ADP is being taken up and ATP is being released. Proton pumps also work with flagella. Here you have your proton pump here, and it's using ATP in this case to drive protons in, and then the flow down will actually cause the flagella to spin. So here's some of the yields. No need to memorize this, although some of you guys probably already know it. But in the cytosol and glycolysis, you have one glucose converted to two pyruvates and two NADHs and two ADPs. And then in the mitochondria, the pyruvates converted to acetyl coenzyme A plus two NADHs, and then the two acetyl coenzyme A's become 6 NADHs, 2 FADs, and 2 GTPs. So the net result in the mitochondria is 2 pyruvates become 8 NADHs with high energy electrons, 2 FADHs, and 2 GTP molecules. And during the ATP synthesis, all of the protons sort of flow through the ATP synthase, and the rotor actually spins, and in this it takes ADP and inorganic phosphate and converts it to ATP. And once again, this can sort of be reversed, where you can take ATP and drive it to establish the proton gradient. So it's a reversible reaction. And they know this actually rotates, and at one point in time, this happened when I was in graduate school. I remember reading the paper where they actually took this molecule, embedded it on a microscope slide, and connected a large rhodamine protein to the end of it, and they watched it spin around using video microscopy, and it looked like a little helicopter spinning around, because there was a big long rhodamine connected to the bottom of it, and it just spun. But the rotation was actually proposed years and years and years before that. So proton movement in all of this is really important, and we've sort of covered proton movement before. So if you just have a bunch of water molecules, the protons can actually jump from one water molecule to the other really easily, and it just creates this environment where protons can move rapidly across different things. One proton is lost, it grabs another one from another water molecule, that one grabs it, and so it's a really sort of a dynamic flux of protons in water. And once again, getting back to oxidation and reduction, here you have your oxidized electron carrier. An electron will come in, produce a transient intermediate, and then you have a reduced form with a proton associated with it. And same thing, if you have a reduced form and you release an electron, and that's eventually going to release a proton. In this case, it can go to water, and then you're basically left with your oxidized electron carrier. And so here's just sort of a model of how electrons move protons. You have your high energy electron. It'll move to one thing, and that'll cause the proton to be taken up. And then as it flows down and loses energy, the proton is eventually released, and then you have your low energy electron. Some of the things that help move electrons are hemes, and there's all kinds of other cofactors, copper, iron. We'll see them also in plants and chlorophyll. There's magnesium. And here's just sort of a schematic of heme. It's got this porphyrin ring here, and with the iron in the middle. And the iron can actually exist in a +2 or +3 state, which makes it really great at moving electrons. And this is just one cytochrome and cytochrome C, but there's all kinds of different hemes. In total, there's 5 different cytochromes in the respiratory chain, and a lot of them have hemes and iron-sulfur cluster proteins, as well as copper. Here's the iron-sulfur clusters. They come in sort of 3 varieties. 2 are shown here. You have a 2 iron, 2 sulfur. So 2 irons and 2 sulfur, and this is liganded by 4 cysteines, and the sulfurs from the cysteines makes this 2 iron, 2 sulfur. You can also have a 4 iron, 4 sulfur cluster where you have 4 irons and 4 sulfurs, and this is also liganded by 4 cysteines. A third type, which I really want you guys to know, and I'll point it out in the upcoming slides, is the risky type. And this is a 2 histidine and 2 cysteine liganded 2 iron 2 sulfur cluster. Okay, so instead of having 4 cysteines here, 2 of them are histidines and 2 of them are cysteines. And this comes into play because of the bifurcated electron transfer that happens in the cytochrome BC1 complex, which I'll show in a couple slides here. Another cofactor that's great at moving electrons is this molecule called ubiquinone, and here it is, and it sits in the membrane, and as you can see here, you have the oxidized ubiquinone, and it can take up electrons, and then that will also take up protons. You have the ubisemiquinone here, which is a free radical, and that'll eventually take up 2 protons. Okay. So it can carry 2 electrons and 2 protons, which is one of the things that makes it such a great carrier. And ubiquinone is also very important in the Q cycle, which we'll see in a second. But here, before we get to the Q cycle that happens in the cytochrome BC1 complex, here are the 3 main enzyme complexes in the inner mitochondrial membrane. You have the NADH dehydrogenase complex, so that's going to take electrons from NADH and leave you with NAD, and in the process it's going to pump protons. Electrons from the NAD dehydrogenase complex actually go to ubiquinone, and then the electrons from ubiquinone go to the cytochrome BC1 complex, and then the electrons are eventually transferred to cytochrome C, and in the process the cytochrome BC1 complex is pumping protons, and eventually the electrons from cytochrome C go over to cytochrome oxidase, and eventually they go down and reduce oxygen into water. Okay? And in the process, the cytochrome oxidase is also pumping protons. And it's really neat how this whole system works, and we're going to look mainly at cytochrome oxidase and the cytochrome BC1 complex, because these enzymes have really evolved fantastically to be able to change the difference in the proton gradient by sort of moving the fewest electrons possible. And we'll see that in a second, specifically in cytochrome oxidase. Okay, so in cytochrome oxidase, in the intermembrane space, you have 4 electrons entering one at a time, okay, from cytochrome C. And they go to this central copper atom and then down to heme A and over to heme B. And eventually they will end up on water or on oxygen. And in the process of sort of adding electrons to oxygen, what you end up doing is pulling out 4 protons and creating 2 molecules of water. Okay, so you're taking up 1 molecule of oxygen, 4 protons, and creating 2 molecules of H2O. But in the process of these electrons moving, they also pump 4 protons across. Okay, so stop and think about this for a second, because what you're doing is you're moving 4 protons plus you're removing 4 protons in the process. And so you can see how just efficiently with just moving 4 electrons, you've had the net result of changing by 8 protons, right? 4 have been pumped and 4 have been taken up and added to oxygen. Okay, so just a really sort of a cool mechanism here with cytochrome oxidase. Here's the structure of cytochrome oxidase. A lot of these electron transport carriers in the mitochondria, they're actually dimers, but in general, electrons just flow down 1/2. Okay, so here's just the 1/2 of cytochrome oxidase where the electrons are flowing, and all of this really doesn't do anything. And I can't answer why. I wish I knew. Maybe somebody knows now, but I don't. Okay, so here's once again the catalytic core. You have electrons coming in from cytochrome C. They go to the central copper ions and then down to heme A, and then heme A3, and then over to heme A, and then they eventually end up on water. And there's also other coppers that are in here, another central copper that's there. Now, one of the things that scientists use to sort of study this whole process is to sort of understand the redox potential, right? Because electrons are always going to sort of flow downhill. And so some of the, NADH is sort of considered the one with the lowest redox potential, and then the electron is going to flow downhill. And you can actually measure these things in the laboratory, the redox potential. And that's sort of how they figured out the electron flow as it sort of goes down here. You don't really have to memorize this. Just really appreciate the downward flow of electrons from a really net negative redox potential down to a more positive redox potential. And here's just the free energy per electron as it's traveling down, right? And there's NAD, and the electrons go from NADH through the NAD dehydrogenase complex onto ubiquinone, and then into the cytochrome BC1 complex, eventually onto cytochrome C, and then into cytochrome oxidase, and eventually onto oxygen to produce water. Okay, now on to the cytochrome BC1 complex. So I've covered a little bit of cytochrome oxidase and how electrons transfer, and I want you guys to understand that, but I don't necessarily need you to memorize it. But I really want you to memorize and understand how electrons move through the cytochrome BC1 complex, because to me it's actually just really fascinating, and I want you to truly appreciate it, because if you understand how electrons are traveling through here and how it's pumping protons, I think you'll get a really good overview of exactly how the system works. Okay. So here you have the cytochrome BC1 complex, and there's three really important players here. Okay, there's cytochrome B, which is shown here in green. There's cytochrome C1 up here, and then there's this protein with this helix here called the risky iron sulfur cluster, or the risky iron sulfur protein. And there you can see it's 2 iron and 2 sulfur cluster, which is liganded by 2 cis's and 2 histidines. And what ends up happening when electrons go through here, electrons come from ubiquinone and they get recycled. And they actually go in 2 different directions. And the risky iron sulfur protein, which has this little neck region right here, actually goes back and forth, okay, to help the bifurcated electron transport. So electrons are going in 2 directions here, okay. One electron is going to go up to cytochrome C1, and one electron is going to go down to cytochrome B. Okay, and we'll see how this happens again. But in the process, and when they crystallize this, they actually use 2 inhibitors. One inhibited down here on cytochrome B, and the other inhibited here. So there's 2 sites where the quinones actually sit, and depending on the inhibitor that they used when they crystallized it, they actually found that the risky iron sulfur protein was in one conformation or down in another conformation. And they think that works as sort of a shunt and a shuttle to basically make sure that one electron is going in one direction and one electron is going in the other direction. So bifurcated, going in 2 directions. And here it is again, a little close-up. You have the risky iron sulfur protein here, and then you have cytochrome C1 with the central heme, and then you have basically 2 hemes in cytochrome B, one BL and then the other BH, and electrons are going in 2 directions. One is going towards cytochrome C1 and eventually to cytochrome C, and the other is going down BL and then down to BH and eventually goes back to the Q pool. And the risky iron sulfur protein is what's sort of shunting the electrons back and forth depending on where they're going. And this creates what's called the Q cycle. And what I love about the Q cycle, and I really want you guys to know this, so listen carefully here, is that there's a recycling event that happens here, right? The enzyme has actually evolved to recycle electrons so that it can actually move more protons with the movement of less electrons. So, and it sort of happens in a 2-step process. Okay, so here's your reduced ubiquinone, and it comes in and it has a binding site, and there's 2 quinone binding sites in the cytochrome C or cytochrome BC1 complex. It's also sometimes called complex 3 or cytochrome C reductase. It's got 3 names. I'm going to go with cytochrome BC1 complex. But here's how it works. You have your reduced ubiquinone, and that has 2 electrons and 2 protons. And so you have, in the first step, 1 electron going in its bifurcated electron transport to the iron sulfur, to the risky iron sulfur protein, then to cytochrome C, and then on to cytochrome C1, and then on to cytochrome C. Okay, so eventually cytochrome C is going to get reduced here. A second electron goes to the heme BL, and then to heme BH, and then it eventually goes on to an oxidized quinone, okay, the ubiquinone here. And in the process, that electron is going to cause it to take up a proton, Okay, so what you see here is one electron goes off to cytochrome C, and that releases 2 protons into the intermembrane space. And meanwhile, you have a proton being taken up from the matrix. Okay, and you have this semi-ubiquinone right here. And this reduced ubiquinone actually becomes oxidized. Okay, the 2 electrons leave, one goes to cytochrome C and one goes down BLBH. Okay, and now you have this semiquinone here, and a second round occurs where another reduced ubiquinone comes in. 2 electrons are released, one goes off to cytochrome C through cytochrome C1 and the risky iron sulfur protein, and the other goes to BL and then down to BH and then ends up on this UB semiquinone. And that pulls up a proton, and the 2 protons here are pumped out. Okay, and so in doing this, ultimately you're with 2 electrons, you're pumping 4 protons and taking up 2 protons, right? So for a net change of 6 protons equivalents, right? 4 are being moved, 2 are being taken up. There's a typo on this slide. This should actually be QH2 here. But I hope you guys appreciate this bifurcated electron transfer and the Q cycle. We're actually recycling the quinones, okay, and using them to drive one round. So with 1 molar equivalent of reduced ubiquinone, you're pumping 4 protons and you're taking up 2 protons, and this is the mechanism by which the electrons travel. Okay. Here's just a model for proton movement. It's believed there's some conformational changes where certain ones are going to favor proton uptake, and then that will cause a conformational change, and then that conformational change causes sort of a drop in affinity for the proton, and it can get released, and then it goes back and relaxes. Okay, simple enough. And here's just a little schematic of showing aerobic versus anaerobic conditions. So if you have the respiratory chain, you'll be pumping protons, and then those protons will then flow through the ATP synthase and convert ADP and inorganic phosphate to ATP. And in the process, take up all of the different things that it needs in the exchangers. And under anaerobic conditions, what you can do is take the ATP and then pump protons out to actually drive the uptakes of these things. Okay, now on to chloroplasts and photosynthesis. Very similar. I'm just going through this so that you guys have a good appreciation of the electron transport chain. So here's just a nice image of the chloroplasts. And here you can see the sort of the granules. There'll be starch there. You can see the thylakoid membranes. And then there's also lipid droplets and a cell wall in here. But here's just a schematic of the chloroplast. You have the stroma and the thylakoid membrane and the thylakoid space. And here it is, and all of the electron transport machinery and the photosynthetic reaction centers, photosystem 1 and photosystem 2, are sort of all in this thylakoid membrane. And so what ends up happening is you basically take in light energy, and through photosynthesis and electron transport, you basically split water into oxygen, and then the high energy electrons and protons from that are converted to NADPH, and it also can, the proton movements that are happening in photosynthesis can generate ATP, and the ATP and the NADPH plus CO2 then enters into carbon fixation, and that converts the CO2 into sugars and amino acids for storage. Okay, so it's basically just the opposite of what we were covering. Here's how carbon fixation works. You get carbon dioxide, and then that will get added on to, if you take that and mix it with this ribulose 1,5 bisphosphate, you get this intermediate, and then eventually you'll get 2 molecules of phosphoglycerate. Okay, so ultimately the CO2 gets added, and then you'll eventually get that converted into sugars. And here's basically how it works. The CO2 is coming in. You have your 3-phosphoglycerate, and that'll use ATP and high energy electrons from NADPH. And ultimately what you're going to do is start to produce sugars and fatty acids. And once again, this happens in plants. People who want to become plant biologists, you should really know this, but I'm not going to hold you to it. So it's just the carbon fixation cycle, right? It's how plants convert CO2 into sugars. Now, the electron transport chain in chloroplasts is somewhat similar to what's happening in mitochondria, except they tend to use chlorophyll. Here's just the chlorophyll molecule. It's got a nice hydrophobic tail and a central magnesium, but it sort of is very similar to the porphyrin ring in heme proteins. Okay, this being cytochrome C here, I believe. And so what ultimately happens is plants in photosystem 1 and photosystem 2 will actually absorb light energy with the chlorophylls. That creates a high energy electron. The high energy electrons eventually will enter into the electron transport chain, as well as you can also steal electrons from water and convert it into oxygen. Okay, that would be the water splitting molecule. And eventually these high energy electrons from photosynthesis get get put on quinone. Here's the structure basically of the photosynthetic reaction center in bacteria. Here you have the cytochrome with these central chlorophylls, and then you have this chlorophyll pair and some other cofactors, and the electrons will absorb a photon of light and cause the electrons to get excited, and then they travel down to the quinone. Here's sort of the look. There's this special pair of chlorophyll molecules, and the electrons basically go from the central pair of chlorophylls, then through pheophytin, and then down to a tightly bound quinone. And that causes it to become reduced. Okay, so here it is. This is very similar to the electron transport chain in mitochondria, except this is using photosynthesis. So it starts off with photosystem 2, which absorbs light energy. You have this water splitting molecule, which will then sort of steal the electrons from water and releases water and 4 protons, and then the electrons eventually go on to quinone. In the case with plants, it's called plastoquinone, and then the quinone actually goes to the cytochrome B6F complex, which is basically identical to the cytochrome BC1 complex that we just went over, and actually has bifurcated electron transfer. The thing is that the cytochromes are slightly different, and eventually, instead of going to cytochrome C, it actually goes to plastocyanin, the electrons, and then the electrons in plastocyanin are delivered to photosystem 2, which then uses light energy to excite them, and eventually goes to ferredoxin, and then gets added to ferredoxin NADP reductase, which then basically reduces the NADP to high energy NADPH, and then this can actually also the proton gradient that's established in this pumping can also be used to drive ATP synthesis. And the reduced NADPH, NATP is then used in the carbon fixation cycle here, where you see the ATP and the reduced NADPH. So that's basically it. So next time we're going to go over apoptosis and cancer. And here's your reading assignments for it. And the reading assignments in the 5th and 6th edition really sort of focus on the apoptosis section and a little less on the cancer. I'm not going to really ask any specifics on the cancer. It's just something I think is a good thing to throw in. You know, the apoptosis lecture is actually pretty short, and then the cancer will just go along with it. Your final exam has been scheduled May 8th and from basically 12 to 3 a.m. That should be 12 p.m. to 3 p.m. You guys aren't here in the middle of the evening. I'm sorry, I typed that wrong. I'll change it before I upload the slides, just so you know it's 12 p.m., so noon, to 3 p.m. On Friday, May 8th. And with the